Okay, this is going to be part one of the amino acid structures, and in this video I'm going to talk about nonpolar amino acids and polar uncharged amino acids. So I drew out the amino acid structures here for the nonpolar amino acids, and I drew most of them out because I thought it would be kind of silly to sit here and have you guys watch me draw these out one by one. So what I just want to do is point out a few things and give you some tips about how to maybe think about drawing these. So the first thing I want everybody to notice is that this backbone here is the same in all of the amino acids, with the exception of proline. And you know, it's really basic. It's just a carbon, a C alpha, and a nitrogen. So the way I think about it is I say, okay, C alpha, N. And for each amino acid, the only thing that's going to change is the side chain. So that's this part right here is basically what's attached to that alpha carbon. That's the only thing that you're going to see change in all of these amino acids. So you can always remember the backbone is going to be exactly the same in each. As far as the nonpolar amino acids go here, the first one I have drawn is alanine. And the abbreviation is ALA. A is the one-letter abbreviation. And the side chain is just a CH3 or a methyl group. The second one is valine, and that's VAL, V as the abbreviations. And the side chain for valine has a CH, a methyl group, and another methyl group. So that's like an isopropyl group, essentially. The third one is leucine, LEU, and L are the abbreviations. And the side chain is CH2, CH, CH3, CH3. The next one over here that I have is isoleucine, and that's slightly different than leucine, and that is we have a CH, a CH2, a CH3 up here, and another CH3 attached to the original CH. So that's isoleucine. Proline is one of the unique amino acid structures. It's a cyclic ring. It's not aromatic though. So it has the nitrogen actually incorporated into the cyclic ring here, as you can see. And these can cause problems in the alpha helix and things like that, because they're pretty rigid. The ring structures are rigid. You probably learned that in organic chemistry. So basically attached to the C alpha we have a CH2, a CH2, a CH2, and an NH. Now I didn't draw these in the Zuider ion form. That's basically the form you would draw them at physiological pH. We're going to do that and go over things like that in the next couple of videos, so I just kind of drew these in the basic format here. The next one is phenylalanine, and the abbreviation is PHE-F, and that does have an aromatic benzene ring. So CH2, benzene ring. Another of the aromatic nonpolar amino acids is tryptophan, and that's TRP-W. And we can see again we have the benzene ring attached here, NH, CH, double bond C, CH2. So that's tryptophan. And the last one I have here is methionine, MET, or M, and that's just CH2, CH2, S, CH3. Now, just to show you guys how I draw these out, I left glycine out, and I'm going to include it in the nonpolar amino acids. So let me just draw that real quick. So again, C alpha, and I almost made a mistake there because this is not a stereocenter. So there's two hydrogens here. There's not four different groups attached to this carbon. So that's one of the unique things about glycine, which is GLY, G as the abbreviations. And it's the simplest amino acid, very small, and can also disrupt the structure. So I don't know if I can get all these on camera at the same time. It doesn't look like it. but those are the nonpolar amino acids. The best tip I can give you for drawing these is to just simply sit down with them and continue drawing them over and over again. A few times, I'd say about 5 times, and you should start to really get the hang of what's going on here. So the second set that I have that I want to show for this video are the polar neutral amino acids, and those are serine, abbreviation SERS, and that's a CH2OH as the side chain. The second one here is threonine, and that's THRT, and that has a CH3OH. The third one over here is tyrosine, TYRY is the abbreviations. That's a CH2 benzene ring, or phenol ring rather, on the side here, or the side chain. And cysteine over here, which is CYSC, which is going to become important later on because that actually forms disulfide bonds. And it has a CH2SH. And I have also over here asparagine, ASN, N, and that has a side chain that's CH2, C double bond O, NH2. So I left off glutamine, and I'll draw that out for you real quick, just again to kind of give you guys an idea of how I'm doing it. Nothing special, nothing spectacular here. So I draw out my backbone, and then glutamine has a CH2, CH2, and then this is a C, double bond O, NH2. Sorry, it's a bit sloppy there, but CH2, CH2, C, NH2. And that's G, L, N, and Q as the abbreviation. So those are the 6 polar neutral amino acids. And again, it's just really practice and repetition that's going to get you good at using these, but you're also going to get used to using them when you start doing protein structure and I ask you to draw a tripeptide or something like that, and you're using a bunch of these amino acids and things like that. So that's all I want to cover for this video, and I'll finish up with the rest of the amino acids in the next video. Thanks.